best greetings from Göttingen Christmas market. It's a photo from last year. Unfortunately, this year there's no Christmas market. And I want to talk about isotopic fingerprints of atmospheric O2 in rocks, minerals, and melts, where I will just give a potpourri on different observations of interaction between O2 and the lithosphere. And so it's something that I think I just want to introduce, and then you should take it and see what can be done scientifically with it. So the atmospheric oxygen, as mentioned in Martin's talk, is by mass the largest reservoir of anomalies with an isotopic anomaly in oxygen triple isotopes. And this is a graph from a review article by Mark Tiemens from 2006 showing here it's called terrestrial fractionation lines. This is a mass-dependent fractionation line, and then all these atmospheric components with an isotope anomaly. So the pathway for atmospheric oxygen is that the photosynthetic oxygen has no isotope anomaly. It's produced, on land, in leaves, and in the ocean. And the O2 produced in photosynthesis basically reflects the isotope composition of the local water, which is leaf water in the case of the terrestrial photosynthesis, and marine seawater for the marine photosynthesis. This oxygen is then fractionated in a mass-dependent way by the so-called Dole effect. It's an effect, I think Dole published this in 1954, and it's the result of respiration of light oxygen by heterotrophic organisms. So it's easier for us to pick up the light oxygen, so heavy oxygen is left behind in the atmosphere. But still, the Dole effect is a kinetic fractionation effect and is mass dependent. But now, from the air oxygen, all these anomalous components are produced, including the stratospheric ozone, which is with the largest positive isotope anomaly. And this has to be counterbalanced by a negative anomaly, a small negative anomaly of the air oxygen that we breathe in. So the air oxygen that we are surrounded with has a small negative isotope anomaly, which Martin showed it's minus 0.43 per mil. And this is a graph showing the isotope composition of atmospheric oxygen of different studies by plotted versus time. And here we have the data for delta 18. So the delta 18 is in the range of 24 per mil. And for the cap delta 17, we see various studies here, and these studies differ in results by as much as around 50 to 60 ppm. But the precision of each measurement is in the range of 5, 6, 7 ppm, so smaller than symbols, which means we are still facing a situation where we can have high precision measurements but low accuracy. And this is currently still a problem when dealing with these very small variations in the triple oxygen isotopes. And this is just a bit a repetition of what Martin already showed, that the definition of the cap delta 17 relative to it's relative to a reference line in the delta 17, delta 18 O space, in the linearized form of the delta space. A number of slopes have been used, and none of this is wrong, none of this is right. And first of all, it's very important, if you write a paper on O17, on high precision measurements, clearly define everything, which slope you're using and which intercept, and how you trace the data back to SMO. This is especially true for O17 because there are currently no standards available for minerals and rocks with a certified small delta 17, or more importantly, a cap delta 17O. regarding, with the reference line slope, well, I have a weak spot for 0.53 or 5 because this has a physical meaning, but in order to get things compatible with the water community, it's, well, I've been, converted to 0.528, which just makes it easier if the water community and the rock and other communities are using the same definition. It's just making life easier. And as I said, there's no wrong, no right value. But this is clear. This is an arbitrary choice of a reference line. It's not the meteoric water line, as pointed out by Martin. The meteoric water line actually does not have the slope of 0.528000, but it's approximately 0.528. But it depends on the samples. It's neither the terrestrial fractionation line, and maybe even not SMO and SLAB are plotting on a line defined by these 2 points that then has a slope of 0.528, which has been, found and published by Vosbrock recently. So it's all, difficult then, to have data from different labs that have an internal precision in the ppm range, but then they may differ from lab to lab on larger scale. Okay, so the Cap delta 17 precision often is in the range of 5 ppm. Accuracy is in the range of 100 or maybe 50 ppm, but this is 20 times the precision, and this is not so good for the moment. But why is this? It's calibration issues for sure. It's on SMO is water, and it's difficult to calibrate rocks relative to water. This is a different extraction procedure. Analytical issues like pressure baseline correction, published in the paper 2018, I think, by Lawrence Young, explaining why we may have problems, gas impurities, and also conceptual issues on some of the papers dealing with high precision or 17 measurements. So there's still a lot to do. Going back to air oxygen, the negative anomaly of air oxygen is minus 432 plus minus 15 ppm, which is an average of recent studies. And it counterbalances positive anomalies of O3 and stratospheric CO2. But this anomaly is diluted by normal oxygen from photosynthesis and then fractionated by the Dole effect. But the Cap delta 17 of air O2, if we would know this through geological times, provides information about pCO2 and gross primary productivity. And these are important biogeochemical parameters. So this is a diagram modified basically after the data published by Ed Young and his colleagues in 2014, where the Cap delta 17 of air O2 is plotted versus the gross primary productivity, the photosynthesis rate, the percent relative to modern. So this is a present day situation, and these gray spots are measurements of air O2. I displayed these in X direction, and we see that there's still some variation, and we should get air O2 better relative to SMO, and then maybe air O2 would be a good standard then to use instead of air O2. It's easier to extract and to calibrate your gas against. So now, how do we get the atmospheric signal into rocks? And one possibility is an oxidation. Oxygen is an oxidizing agent, and it has been shown that you can oxidize a pyrite and form from this sulfur form sulfate, and this sulfate contains atmospheric oxygen. And if this sulfate then is precipitated, you get information about the isotope anomaly of atmospheric oxygen. Other approaches use bioapatite and carbonate. Here is as a skeleton or eggshells, and bioapatite and carbonate precipitate from body water, and the body water mixes with metabolic water and CO2 from the oxidation of nutrients. This is how we get our energy. Then deep sea manganese nodules have been shown to have the isotope anomaly of of air oxygen. We can also have high temperature exchange, and I will show examples on Tactiles and cosmic ferrules. For the cosmic ferrules, it's both, it's high temperature exchange and oxidation. And in my book chapter, I'm also showing data on technical products that also have exchange with air oxygen, but I'm not showing this in this presentation today. So for the sedimentary sulfate, it's Hu Min Pao and his group first discovered that some of the sulfates here with an age of about 650 million years have a strong negative anomaly in Cap delta 17. And they correctly traced this anomaly back to the other component with a negative anomaly, or the only component with a negative anomaly that we know, and it's air oxygen. So this sulfate from sediments, barite sediments, this has inherited the isotope anomaly from oxidation, subaerial oxidation of pyrite with O2, and the sulfate is then transported in rivers, transported in seawater, and then precipitated in closed basins with a lot of evaporation, and then this barite can precipitate. And these sulfates preserved the isotope anomaly of the air O2 at time when they formed by oxidation of pyrite. And the anomaly goes down 700 ppm, that's a lot. And Bao et al. Attributed this to more than 10,000 ppm of CO2 in the atmosphere, which could have accumulated during the snowball Earth event that occurred immediately before these deposits have formed. And this is the first, to my knowledge, the first paper on traces of atmospheric O2 in rocks. Recently, Peters et al. Published a paper on magnetite that also has an isotope anomaly. This is a diagram Cap delta 17 versus delta 18. And this gray area here is where we expect, all the crustal rocks and minerals. And they analyzed magnetite, and this magnetite clearly shows a negative anomaly. And these are magnetites from apatite magnetite deposits. And the chain of argumentation was that pyrite and O2 reacted to form sulfate with a portion of anomalous oxygen in the sulfate. This sulfate then exchanged with fluids, maybe by sulfate reduction. These fluids carried an anomaly and then imprinted this anomaly to the magnetite. And this demonstrates how persistent these anomalies are. So we have a long chain of exchange, including fluids, but still the anomaly is preserved. Another oxidation product is the oxidation of nutrition. This is a sugar, and if I take this and oxidize it with O2, then I gain energy, and the reaction product is CO2 and H2O. These two mix, the metabolic water and CO2 mix with body water, and it has been shown by a very nice study by Whiteman et al. In 2019 on measurements of body water that actually you can see the isotope anomaly in the body water of vertebrates. And these data points here, you can see that this is plotted versus the body mass, and the smaller the animals, the larger the portion of anomalous oxygen in their body water. And this can be explained by the high specific metabolic rate of small animals compared to large animals. And they concluded by a simple mass balance model, this is a composition of atmospheric oxygen, and this is a composition of the drinking water, that this small mouse contains something like 54% of oxygen in the body water directly coming from air oxygen. And this is very close to the theoretical limit of 66%, which is 2/3 of the oxygen in case that this animal is not drinking anything. And similar observation has been made earlier in a paper that we published in 2013, where we have the body mass plotted versus the cap delta 17 versus body mass, and we can see a broad relationship between body mass and cap delta 17 of the appetite. So we analyze the bioappetite bones and teeth, and here you can also see the anomaly. So fossil bones and teeth would carry the signal of the isotope anomaly of air oxygen. And same is true for carbonates. Ben Pesce and his colleagues, they published a paper in 2014 on triple isotope measurements of carbonates, which is it's very difficult to analyze the triple isotopes of carbonates. Either you do it by fluorination in bombs, and we tried this, couldn't get it running, and Ben Pesce developed a very complicated protocol to analyze the triple isotope composition of carbonates, and results are shown here. So the reaction is that we have the sugar plus air oxygen or inhaled oxygen reacts to CO2 H2O, mixes with body water, and then precipitates 39 degrees is the temperature of what birds have into calcite of eggshells. And this plot of the cap delta 17 versus delta 18 shows the range of abiotic carbonates measured at this time. And here we can see that the vertebrate carbonate, the eggshells, have a more negative anomaly, and this negative anomaly stems from intake of anomalous air oxygen, but it's a bit more complicated here. These are I think desert ostriches, or so desert animals that also drank highly fractionated evaporative water that is also going down because the slope for the evaporation is lower than 0.528. But certainly there's also an atmospheric signature in these eggshells. Then recently, Sutherland et al. Published a paper on deep sea manganese nodules. And there was a paper in the 90s by Mandanak et al. Who demonstrated that, that apparently manganese 4 oxides, preserve the isotope signal from O2 that is needed to oxidize the manganese to the manganese 4 plus. And this is a diagram of cap delta 17 versus delta 18 with the data of Sutherland in here. And it's clear that there's a negative anomaly in the manganese oxides, and this anomaly is inherited from dissolved oxygen in the seawater that eventually came out of the atmosphere or equilibrated with the atmosphere. And so deep sea manganese nodules and crust are potential archives for the isotope anomaly of air oxygen. But as it's shown here is that to trace the composition of air oxygen back from the measurements of manganese oxides is, at this moment, it's very difficult because many reservoirs can be involved here. Another component that carries or can carry an isotope signature are tactiles, and tactiles are silicate melts that form in a meteorite impact. And these tactiles are composed of, it's nearly pure target material. So it's if any, very little meteoritic component in it. So it's you can assume that it's a target material. So it's Earth crust. And this Earth crust material is melted, flies through the atmosphere, and then at temperatures exceeding something like 1800 degrees Celsius, the air O2 exchanges with the silicate melt. And we have done exchange experiments, and this is a starting material that we melted at 1350 and 1500 degrees Celsius. This is a cap delta 17 plotted versus delta 18. And this is a composition of air O2. And what we observed is, and the sample size were in the range of 1-2 millimeters. And what we observe is that already after 30 seconds or so, we had something like 50% exchange with air oxygen. And at high temperatures, 1350, 1500 degrees, we still had a mass fractionation between equilibrium fractionation of 2.5 per ml between O2 and silicate melt. So now looking at data on tactiles, these are data from a paper by Thomas Magner from 2017 and Karel Zack and colleagues from 2019. And on tactiles. So most tactiles have the composition of crustal rocks, very narrow range here. But some tactiles, these are the so-called ergosites from an impact site in Kazakhstan, they seem to have exchanged with air oxygen. And what I plot here now is that this is the equilibrated melt point, and then we have mixing with the crustal material. The Clayton data here, probably the error bar is just too large really to decide whether they have an anomaly here. And this would be the trend towards the carbonaceous chondrites. The last objects that I want to show are so-called cosmic I-type spherules, and these form by oxidation of extraterrestrial iron nickel in the atmosphere with O2 at 80 kilometers. Maybe also some oxygen is photolyzed and occurs as oxygen atoms. But this bulk atmospheric oxygen then oxidizes these alloys to wüstite and magnetite. And these are images here of these cosmic spherules. And the exciting thing in these cosmic spherules is that they have 100% air oxygen. Unlike all the other objects and materials that I've shown where a fraction, and the problem is often an unknown fraction of oxygen is from the atmosphere, here we know it's 100%. And these are, I think, good proxies for the composition of the atmosphere. And these are data that is a paper by Michael Fisher. It's in revision at the moment. And these are data on the triple oxygen isotopes kept delta 17 versus delta 18 of Antarctic I-type spherules. And these spherules plot on a relatively tight trend in the triple isotope diagram. The slope in this trend has not a single physical meaning, because this is a mixture of fractionation during oxidation, fractionation during evaporation. You see they are pretty heavy in delta 18, and fractionation and equilibration exchange with ambient oxygen. So our approach now is that we just fit in a line through these data, and then assuming that we would have an atmosphere with 800 ppm CO2, the air O2 would be here under the precondition that the GPP is the same, and then these cosmic spherules would lie here. So if we find cosmic spherules in the rock record and analyze them, one cosmic spherule could tell you about the PCO2, or first of all about the kept delta 17 of the atmosphere, and from this then about PCO2 GPP at time of when they came down. Okay, well, to sum it up, first of all, more labs need to calibrate the tank O2 relative to small water and then publish data on available standards. It's still the agreement between laboratories is I think it's becoming better, but it's still not good enough. We should really have data of San Carlos olivine or UWG garnet, let's say in the range of plus minus 5 ppm. This is what the precision gives, and this would be very helpful. So if you go into the O17 field, give you a kick and say, okay, well, I also want to fluorinate some water. It's a pain, but it's doable. And then try to calibrate your tank O2 and rocks and minerals yourself using water from IAEA in Vienna. And the kept delta 17 of O2 or traces of it can be preserved in anomalous sulfates from subaerial pyrite oxidation, transferred to magnetite. So also these magnetites can carry an anomaly. It can be preserved in fossil vertebrate, bioapatite, and biocarbonate. It can be found in deep sea manganese nodules and crust. Tactiles can carry a signal from the atmosphere and cosmic I-type ferrules, and also technical products, alumina, artificial gemstones, which I show an example in the book. Good. And well, it's with this I come, to the end of my talk. I thank very much Ilya Bindeman for initiating, this book project and eventually also, this short course, and Benbow and the MSA staff for their really valuable support. And I want to thank my group from Göttingen, which is a picture from last year. Okay. Thank you very much.